Hello everyone, welcome back to the channel. In this video, we will discuss NCBI BLAST, one of the most important bioinformatics tools used in biotechnology, genomics, and proteomics. If you are a B.Sc, M.Sc, or research student, understanding BLAST is absolutely essential. So let us begin. BLAST stands for Basic Local Alignment Search Tool. It is developed by NCBI and is used to compare a DNA or protein sequence with sequences stored in public databases. BLAST performs local alignment, meaning it finds regions of similarity, not necessarily the entire sequence. This makes BLAST fast, efficient, highly reliable. BLAST is widely used because it is user-friendly. BLAST works through a simple web interface and does not require advanced coding skills. It is extremely fast. It can compare your sequence against millions of sequences within seconds. It searches curated databases. BLAST uses high-quality databases like GenBank, Besic, PDB. It helps in functional annotation. By finding similar sequences, BLAST helps predict gene function, protein function, evolutionary relationships. KeyCap1 nucleotide BLASTn is used for DNA or RNA sequence comparison. It compares a nucleotide query sequence against a nucleotide database. Applications of BLASTn: gene identification, cloning verification, mutation analysis, phylogenetic studies. Pushpin example: if you clone a gene and want to confirm whether it matches a known gene, you use BLASTn. KeyCap2 protein BLASTp is used for protein sequence comparison. It compares an amino acid sequence against a protein database. Protein BLAST is more sensitive than nucleotide BLAST because protein sequences are more conserved during evolution. Applications of BLASTp: predicting protein function, identifying conserved domains, studying protein evolution. Let us understand this with an example. Imagine you isolate a bacterium, sequence one of its genes, translate it into a protein of 312 amino acids. Now the problem is, right finger, you do not know the function of this protein. So you upload the protein sequence into BLASTp. How to interpret the BLAST result after running BLASTp? You observe high sequence similarity, very low E-value, high query coverage, conserved protein domains. What does this mean? A low E-value means the match is not random. High identity indicates true homology. Conserved domains suggest same molecular function. Pushpin: based on this, you can confidently annotate your protein. For example, this protein is a DNA helicase involved in DNA repair. Interpret BLASTp results. Let's start the explanation. For example, I go to the RCBS PDB protein data bank, and for example I took beta-galactosidase, which you can simply search it. It is the website and looks like this. Now enter the first search result. You can easily access the organism, classification, it is a E. coli protein, deposited author name, and other things. If you are interested to analyze the RCBS PDB in detail, then comment it. I will explain it in another video. Then click in the display file and download the FASTA format, or you can easily download the PDB file of it. As I download the FASTA format, it looks like this, and you can open it in Notepad or Word. Now come to the NCBI BLAST, protein BLAST. It is the website interface. Just look it once and ensure the seeding is all in default. Now paste the FASTA sequence or upload the PDB file. Now see, what's are the default algorithm. Yopu can see here Blossom 62. If you want to know it in detail, I will explain you. Just comment it, and look other default parameters. It took little bit of time, 2 to 5 minutes. Be patient, and now it's your result is here. Let's understand each parameter first. Max score. The max score is the highest alignment score obtained for a single high-scoring segment pair, HSP, between the query and a database sequence. It reflects the strength of the best local alignment region. Total score. The total score is the sum of scores of all aligned segments between the query and the subject sequence. It represents the overall quality of the alignment across the sequence. Query coverage. Query coverage indicates the percentage of the query sequence that is aligned with the subject sequence. High query coverage means most or all of the query sequence is included in the alignment. E-value. The E-value represents the number of alignments expected by chance with a similar score in a database search. Lower E-values indicate more statistically significant and reliable matches. Identity. Identity refers to the percentage of exact matches between amino acids or nucleotides in the aligned region. High identity suggests the sequences are the same or very closely related. Similarity. Similarity measures how many residues are chemically or functionally similar, not necessarily identical. It accounts for conservative substitutions that maintain protein function. Accession number. The accession number is a unique identifier assigned to each sequence entry in a biological database. It allows precise retrieval and referencing of that specific sequence record. When we analyze BLAST results, it is very important to understand what the different BLAST parameters actually tell us about our sequence. One of the most important parameters is the E-value. The E-value, or expectation value, indicates how many times a similar alignment could occur purely by chance when searching a database of a particular size. When the E-value is 0.0 or extremely close to 0, it means that the alignment is highly significant and not random. In practical terms, this tells us that the similarity between our query sequence and the database sequence is real and biologically meaningful. Another important parameter is the bit score. The bit score represents the quality of the alignment by taking into account both the alignment length and the number of matching residues. A high bit score indicates a strong and reliable alignment. Unlike raw scores, bit scores allow comparison between different BLAST searches, making them a standardized measure of alignment strength. The percent identity tells us how many amino acids or nucleotides are exactly the same between the query and the subject sequence over the aligned region. When the percent identity is very high, for example around 99 to 100%, it strongly suggests that the sequences represent the same protein or very closely related proteins from different strains or organisms. High percent identity is especially important when confirming the identity of a known gene or protein. Query coverage indicates how much of the query sequence is included in the alignment. High query coverage, such as 98 to 100%, means that almost the entire query sequence aligns with the database sequence. This is crucial because it shows that the match is not limited to a small conserved region but represents a full-length alignment, increasing confidence in the annotation. Finally, the cluster size seen in BLAST results reflects how many similar sequences are grouped together. A large cluster size indicates that the protein is widely distributed, well-studied, and evolutionarily conserved. Proteins with large clusters are often essential for basic cellular functions and tend to show little variation across organisms or strains. Together, these BLAST parameters allow us to confidently identify a sequence, assess the strength of the match, confirm functional annotation, and understand how conserved the protein is across evolution. This is why proper interpretation of BLAST results is just as important as running the BLAST search itself. This image summarizes the protein BLASTp analysis of beta-galactosidase, LACH, from E. coli. The left panel shows that the query protein is correctly identified as beta-galactosidase, supported by very high percent identity, approximately equals 99 to 100%, high query coverage, 98 to 100%, and an E-value of 0.0, indicating a highly significant match. The right panel explains the molecular function of the protein, where beta-galactosidase catalyzes the hydrolysis of lactose into glucose and galactose. The presence of thousands of similar sequences across databases highlights that LACH is a well-conserved and essential metabolic enzyme. The bottom section explains the significance of BLAST parameters, showing that high bit score, high identity, and large cluster size together confirm a strong, full-length, and reliable protein match. BLAST is a core bioinformatics tool. BLASTn is used for DNA. BLASTp is used for proteins. BLAST helps identify gene function, protein function, evolutionary relationships. Understanding BLAST is essential for genomics, proteomics, and research projects. If you found this video helpful, please like, share, and subscribe to the channel. If you want a step-by-step BLAST tutorial or a real BLAST result explanation, comment below. Thank you for watching.